the first point I'd like to make is that whilst loads of people will tell you that horse nutrition is an incredibly scientifically based industry, I don't happen to agree with that. I think there's very little true science in horses, and the people I'm most sceptical of are the people that say their products are scientifically proven. My products aren't scientifically proven. I'm going to show you the evidence that we have for why they work, and you can make your own judgments on that. We have a number of specialities, but the speciality I'm talking about today is primarily about chelated calcium, and I'll explain to you what I mean by that in a minute. What we like to think we try and do is complete this triangle up here. So we're selling you products that are based on nutritional products related mostly to calcium and magnesium. We like to get loads of feedback from people about how the animal's functioning. So when we're talking about calming supplements, we're talking about behaviour. And then we like to see what the real scientists have to say, because, as I said, there's very little equine science. So we dig into the biochemistry textbooks and the neuroscience textbooks and the physiology textbooks. We're trying to get an understanding of what's going on. I'm going to start, and please indulge me here, because in a way I'm not actually today going to present you with evidence to support these things, but I'm going to give you a long list of things that chelated calcium helps with. And like I say, I will explain the difference between the calcium you've probably been feeding your horses for decades and chelated calcium in a minute. To start with, chelated calcium seems to help. This is a sort of generic part of this. Any organs that use calcium cellular signaling. Now, don't worry what that is. I'll come to that in a minute. It seems to help most horses. Now, I guess we're selling a lot of product in the UK. We're selling a little bit of product in America, and we're beginning to sell more and more here in Australia. And we don't see any huge differences in the way the products respond. Bizarrely, and this sounds a bit strange, but it seems to help horses that are already grazing high calcium pastures. We actually developed our chelated calcium technology on horses in the Cotswolds of England, which are solid limestone. There's limestone lying around all over the tops of the fields. And finally, more important for Australia, horses eating oxalate-based pastures, so Queensland, northern New South Wales with buffel grass and whatever. But surprisingly, I think we're beginning to learn that there are issues in a lot of other. The further south we come, we're still finding issues relating to oxalates. Okay, let's look at it now from another point of view. I guess our starting point was always behaviour. So horses that are nervous, spooky, horses that are struggling with concentration and focus, horses that are making bad decisions. That probably applies more to jumpers than to dressage horses. Horses that can't concentrate on their rider properly, so they're not trained, not so easy to train. And horses showing stereotypical behaviours like weaving, box walking, crib biting, wind sucking, those sorts of things. If we look at get a little bit more technical into the areas that we seem to be affecting that cause that previous list of things. Neurotransmitters are chemicals that pass a message from one nerve cell to another, and they are controlled by calcium signaling, and calcium has an effect on them, and chelated calcium seems to improve the release of neurotransmitters. The reception of some neurotransmitters, I must emphasise some neurotransmitters, is also calcium dependent, and so we appear to influence that. I've got a question mark on the next one. I'm not sure about this one yet, but I think when we talk about things like Cushing's disease, the reabsorption of neurotransmitters like dopamine may also be affected by this. Jumped ahead of myself, haven't I? Dopamine gets a mention and Cushing's gets a mention. Now, nerves are not the only things that use calcium signalling. Muscles use exactly the same biochemical technology. And in fact, when you pump calcium into a muscle cell, it contracts. And when the calcium leaves the muscle cell, it relaxes. And we see a lot of issues where horses tie up. And one of the common causes of tying up, it's got very rarely got anything to do with lactic acid, which is the favourite myth around there. It's mostly the lack of the ability to get the calcium out of the muscle cell. And if you can't ship the calcium out of the muscle cell, then the animal's muscles will stay contracted. We know that calcium signaling is involved a lot in heart function. That's not an area we've studied in any detail. Interesting one, this one came up this morning actually with a customer, a non-customer in Queensland. Beginning to go through a list of all the different cell types that use this calcium cellular signaling technology, and sweating is one of them. So where nerve cells are releasing a complicated chemical like a neurotransmitter, sweat cells are releasing water. But the switch to switch it on is controlled by calcium. And we anecdotally have reasonably good evidence of treating horses that don't sweat. It's not a major problem that I know of here, but I suspect the further north we go and the more humid it gets, the worse it'll get. And we certainly find a lot of horses in America, particularly Florida, that suffer this problem. Tendons and ligaments are an interesting one. I always used to think of tendons and ligaments a bit like a very, very solid rubber band. But actually they're quite dynamic. They don't stretch very much, but they're a dynamic, living organism. And they're primarily made of a chemical called collagen. And hey presto, the release of collagen is controlled by calcium signaling. Hoof and hair production. The hooves and hairs are made of a protein called keratin, and again, keratin is controlled, or the release of keratin is controlled by calcium signaling. And to give you an illustration, because my wife's in the audience and she always wants to be mentioned, or aren't I cruel? I know when my wife gets chelated calcium deficient because we'll be driving along the road and a car will come the other way and she'll jump out of her skin and tell me I'm a useless driver and I really ought to do better. She will know that she gets calcium deficient because her fingernails start breaking. And since your horse spends its time standing on 4 fingernails, this is crucially important. So certainly not saying that chelated calcium is the only important thing for your horse's hooves, but it is one of the important things for your horse's hooves. If sweat and collagen and keratin and neurotransmitters are controlled by calcium signaling, so are hormones. We have a lovely video, if you look on our YouTube channel, on a Bolshe lamanitic mare who showed us both of these. Her sex hormones became well regulated when she went onto a chelated calcium supplement, and she had a bout of laminitis at the time, which cleared up within about 3 days. And what we've seen later, again purely anecdotally, is that horses with sex hormone issues, so very marish horses, a number of stallions tend to calm down nicely, and I'm glad to say a lot of horses that are probably on Regumate now, either for breeding purposes or just to enable them to be controlled, often won't need that sort of intervention. And the last one, I guess, is the one that appears not to be a surprise because we all know that calcium is important for teeth and bones. Interestingly though, the chelated calcium is not supplying the mineral that goes into the bones. What we believe is happening is these 2 cells called osteoblasts and osteoclasts. Typical biologists come up with 2 names that are so similar you wouldn't have a clue, and I can never remember which one does which. One of those pumps the calcium into the bones and the other one pulls it out and sticks it into the bloodstream. These are absolutely vital for the regulation of calcium in your horse, and they're switched on by calcium cellular signaling, and that's where we believe our chelated calcium is operating. It's not supplying the calcium that goes into the bones, it's helping the bones to regulate, helping the bones to be regulated, the blood calcium level to be regulated. Loads of horses have ulcers, horrible problem, and this is certainly not the solution to, not a treatment for ulcers, but the release of hydrochloric acid into the stomach is controlled by calcium cellular signaling. So here again, our anecdotal evidence from, I'm glad to say, a number of veterinary surgeons is these horses that get treated for ulcers by drugs like Gastrogard. Is that Gastrogard the one you use here? Whatever. And then they come off the drugs and they relapse and they get bad again. When the horses go on to our chelated calcium supplements, they normally don't relapse. And we originally thought that that was simply because we're giving them a calmer that de-stresses them. I now believe it's that we're helping them to control the release of the hydrochloric acid into the stomach. And that a lot of ulcers, although they may be stress-induced in the first place, that whole release process goes out of kilter. And an exciting one that I can't substantiate at all, but I think is potentially very interesting for the scientists that look at these things, is actually whether a gene is switched on or off is often controlled by a calcium switching thing as well. And I think we're now learning just how much environment affects which genes are working. And this is going to be a really exciting area of scientific research, which is probably way beyond my pocket to be able to fund. Okay, so where are we coming from? I said there's very little science in our industry, and I passionately believe that. So where are we getting the information that I'm going to present to you? Firstly, because we primarily run our business as a mail order business, we are truly intimately connected to our customers in a way that selling a product through a retailer will never ever achieve. And I guess because we would love to know more about how our product works as well as anything else, and we are passionate about getting a good result for our customers. We put a lot of effort into monitoring, getting feedback from customers, and I believe that if we fail, if we sell you a product and you phone me up a month later and you say it hasn't worked, it's rubbish, if you work with us, then we learn so much more from our failures than we do from our successes. And we are passionate about that. And that's why we haven't just sold to 4,000 people. We've got a database of 4,000 horses that have gone through our process. Now, some of that's a few years ago, and we do things very differently now. But it's a very important source of information for us. Blood trials. What really informed yesterday's talk on magnesium was blood trials we ran to try and understand what's happening in the chelated calcium area. We've run a number of blood trials on a number of horses. I know 33 horses is not a huge number, and I don't pretend it is. 5 of those are in Australia. Those horses are a totally different data set because they all had big head related problems, but we still have a huge powerful set of information. I would not recommend any of you do a blood test on your horses. This has been a useful research tool, but it's generally very expensive. It may tell you something about your horse's magnesium. It's going to tell you nothing useful about calcium. And I might explain why later, but don't waste your money on blood trials. Your vet almost certainly, by the way, has never done more than a total blood calcium result, and they're always in the normal range, even on seriously calcium stressed horses, and he's almost never done a magnesium test. So your vet's unlikely to have a clue how to interpret the information they get. Literature review, that's the bit really about how we try and go to the fundamental science. I'm I'm a bit lazy. Looking through a whole bunch of scientific papers is a seriously boring and time-consuming process. So I rely on textbooks, and I reckon that if a textbook's sold or used by a university student, it's probably good enough for me. So my literature review tends to be restricted to, not completely to textbooks, but largely. And the last one there is the only independent study we've had done on our supplements thus far. And Jane is an epidemiologist with the University of the West of England who ran a small trial on some dressage horses, some show jumpers and some eventers, and got some spectacular statistical results out of it that demonstrate what our products tend to do both for performance and behaviour. So that was very useful, and it's independent. This process, lumping all this together, may sound fairly logical, and it seems to me to be very logical, but actually I think the way we leverage all of our customers into effectively our R&D process is actually quite unique. I don't really know anybody else that's doing it. And we did this year, and we're quite proud of this. We won the Business Innovation of the Year award by the Federation of Small Business in Great Britain. Now, you may say, oh, it's only small businesses. Small businesses up to 250 employees. We're a lot less than that. That would actually catch every single feed company in the UK. So I regard them as pretty big businesses. So we're very proud of that. Okay, now then, I want the people that came to yesterday's lecture to shut up now, and I'm going to ask the rest of you to answer this question. How many people think that horses should be naturally spooky and nervous? Come on, put your hands up. Only 4 or 5, 7, 8, something like that. Okay, who think they shouldn't be? The word has spread, hasn't it? Okay. Are those lions hunting?
Sorry?
Are those lions hunting? No, they're not, are they? Are the gazelles running away? No. Prey animals that evolved on the open plains of Asia don't have a tree to hide behind unless they can run 1,000 miles. So, spooking at a flapping leaf or a butterfly, which most of us have sat on a horse and they've done it, is not really very survival oriented. It's not something that evolution would have worked at. I passionately believe that horses should be good risk assessors. And the reality is, most of our horses are rubbish risk assessors. Because if you're going to spook at that butterfly, or worry about what the mare's doing 100 yards away, and you can't concentrate on your rider or whatever, you're clearly not assessing the risks in your environment. Not many of your horses have ever seen a lion, but they seem to worry about where that lion is. You know, ginormous lions hide under whiteboards, don't they? And behind flower pots, all sorts of things. So the second part of this really is if a horse has to be a good risk assessor, then can sedating it improve its brain function and improve its function? And I would argue that it can't. So if we're talking about calming horses down, you really have a choice. Choice A is to sedate it. And people phone us up, I don't want to sedate my horse, I just want you to take the edge off it. Okay, you want me to sedate your horse, I can do that. That's easy. I'll pump it full of magnesium, you can get some ACP from the vet, there are loads of ways of sedating your horse, that's not a problem. And I have to say, for a lot of riders, idiot useless riders like me, a sedated horse feels a lot more comfortable than an unsedated horse. And sedating a horse can be quite a good idea, although those of you at yesterday's lecture will know that I also believe that you sedate a horse, you make its ability to assess risk even worse, you put it under pressure, it may explode and be far more dangerous. So, what we are all about exclusively is trying to get the brain to function as well as nature intended it to. Enable your horse to recognise that there isn't a lion behind the flower pot, to make a really good decision. I mean, if you're show jumping, you walk the course, don't you? And you pace out all the distances, and you know exactly, well, some of us get lost, but basically you know where you're going, and your horse has no idea. You confront your horse with 3 fences, all that it thinks it might jump over. It's got about 3 or 4 or 5 paces to work out how high the fence is, how far away it is, how wide it is, and yes, whether there is a lion hiding behind the wings. And it's got to make all of those decisions really quickly. And if its brain's not working properly, it won't do it. Okay, so let me tell you a little bit about what we have done. I showed this picture before. I said I like looking at textbooks. I particularly like the name of this textbook, which is, sorry about the technical stuff, The Role of Calcium and Comparable Cations in Animal Behaviour. Given that when we started selling calcium-based calmers, all of my competitors said, nah, can't work. I think this is just a lovely title. It's published by the Royal Society of Chemistry, so I think it has a reasonable amount of credibility. It's just one of the books that I like reading. My copy is well thumbed. Oh, sorry. I showed these figures yesterday. We're now looking at the amount of certain minerals inside the cell versus the amount outside the cell, just in the fluid around the cell. So sodium, I don't know why do we call it Na? Natrium, thank you. There's 15 times more sodium outside the cell than inside the cell. Potassium, kalium, there's 30 times more inside the cell than outside the cell. And it's well understood how these sort of work as a pair, and they flick across the cell membrane when messages are being sent around cells. I'm going to compare that with everybody's favourite, magnesium. Magnesium is incredibly difficult to control. You've only got to read the basic biochemistry textbooks, and whilst they all say the theory is, or the hypothesis, so nobody's actually researchers, and now I'm talking about the world of human and biological physiology and science and whatever, but the theory is that magnesium ions are so tiny, in order for a protein to grab hold of it, the protein has to bend more than it wants to go. That's the theory. So it's actually, imagine trying to pick up a ball bearing with a set of gardening gloves on. It's really difficult, and that's the theory. So magnesium is difficult to get hold of. If you can't get hold of it, you can't push it around and move it across membranes or whatever. So magnesium, which is desperately important, everybody tells you it's got 300 jobs. None of them involve it being really precisely controlled. And therefore, there is roughly the same amount of magnesium inside and outside the cell. Some of them will be 2 to 1 or even 3 to 1. So it's not totally uncontrollable, but it's not dramatic. Anybody like to take a punt at what those figures are going to be for calcium?
Do we have to shut up again? Yeah.
Yes, you have to shut up again. No, go on. Look intelligent. You know, whatever. 30,000. Oh.
Never mind, we've got it down here.
Scary. Yeah, 30,000. That's not a lot of calcium outside the cell. That's a very, very low amount of calcium inside the cell.
Ah, so it's, the left side is outside of the cell, and the...
well, actually, I think I had that wrong before, didn't I? You know, that's the right way round. I gave this presentation to the Royal Agricultural University, and some smart-ass student observed that I'd got those numbers the wrong way round. They are the right way round now. So there is very, very little calcium inside the cell.
Yeah. Oh, inside.
Okay. And that is the huge investment that your body makes and your horse's body makes, basically in brain power, because it's the brain that's doing this a little bit more than everything else. So we humans, I can't remember, human brain is 2% of our body weight, I think, 1% is something like that. It's 25% of our energy consumption. And a lot of that energy is pumping calcium ions out of the cell all the time. So, This is my representation of a nerve cell. It's bathed in a fluid that's rich in calcium, and although this is a gross oversimplification, the white bit is the calcium-free zone within the nerve cell. And when we want to switch a nerve cell on, what we do is we just pump a little pulse of calcium ions in, and that basically roams around, finds these switches, these calcium receptors inside the cell, and flicks them on. So it's just like you walking into a room, turning the light on. Really, really simple. And that's how it does it. Calcium ion sits on it and flicks the switch. The point to note here is, contrary to what most people supplying you with magnesium products will tell you, there is no role for magnesium in this. It just isn't involved. And whilst we understand that those sodium and potassium work as a pair, 40 years ago the theory was that calcium and magnesium would work as a pair. And that was a pretty good theory. The trouble is that the neuroscientists and the physiologists have done their work and it doesn't happen to be true. So magnesium is not important in switching cells on, or, as they used to tell you, calcium's the evil stuff that goes in and excites the cell. We biologists have a horrible habit of using emotive words for simple things. So calcium's exciting the cell. Magnesium apparently went in and pushed it all out and relaxed it. That isn't true. It just simply doesn't happen. Just to show that I do read more technical, that's just an example of a neuroscience textbook, what these channels look like. They don't really look like that at all. Okay. I talked about doing some blood trials. We started them in late 2011. And when we started, we believed what the veterinary textbooks will certainly still tell you, that the only biologically active form of calcium is the calcium ions, and they are the ones that go into the cell and switch it on. So they are clearly desperately important. Our trials suggest that that's not correct. But we believed it, and in fact the first blood test we did, we only tested for ionized calcium, because that's what we thought we needed to look at. So let's look at the forms of calcium actually inside the blood. This is textbook stuff again. 35% of it is bound to protein molecules. We now, when we do a blood test, we measure the amount of protein in the blood, and particularly a protein called albumin, because that binds loads of calcium. And you can vaguely guesstimate with some formula that somebody invented a long time ago how much calcium is bound to the proteins. That is apparently not biologically active, and because all the horses we've blood tested haven't shown big changes in protein, I don't know whether I agree with that or not, but I suspect it's true. It's just sort of dormant calcium not doing anything. The next bit is the ionized calcium. So this is, your horse will get this out of its grass, out of the cereal grains you may be feeding it. It'll get out of the limestone that your feed company, or the dicalcium phosphate that your feed company puts into formulated feeds. And I'll show you later on, I think, what I mean by ionized. Oh, there we go. That's what I mean. That's calcium carbonate. And when calcium carbonate dissolves, what happens is it splits. A couple of electrons fly from the calcium to the carbonate bit, and it becomes negatively charged, and you see there are two negatives there. And the calcium, because it's lost two negatively charged electrons, becomes positive. And that is the active calcium ion that goes into the cell and flicks the switches. The next group are complexed calcium. And the textbooks don't talk about this because the text they mention it's there, but they regard it as inactive. I split it into two, so I'm deviating from the textbooks here. I've got what I call inorganically complexed. That's things like calcium bicarbonate. So that's stuff that dissolves but doesn't split up. It stays as a largest molecule. and you've got the organically complex ones. Your vet will routinely use calcium gluconate. Glucose molecules joined to calcium produce calcium gluconate. That's an organically complex one, or a chelated calcium, if you like to use that word. It's that one. That is calcium gluconate. You can see the calcium in the middle, and it's joined by hydrogen bonds to what started off as glucose, and those sharp-eyed ones of you who know your chemistry know that glucose has got a ring structure in it. That ring structure actually breaks down in calcium gluconate, but you've got big organic molecules either side of the calcium. This one's a much more complicated one. This is calcium citrate, and here you've got a calcium ion here, a calcium ion here, and a calcium ion here. So you've actually got 3 calcium ions and 2 citrate molecules, and this one's really good because you actually get 4 water molecules attached to it as well, which they don't show exactly where they are. But, so we're talking a fairly complicated and large molecule here. And this is the one some of you guys will have a challenge with. This is calcium oxalate. Calcium oxalate is also a chelated calcium, but it ain't a good one. Calcium oxalate causes all sorts of problems. But again, you can see the calcium ion, and it's hydrogen bonded. It doesn't show it there. It's hydrogen bonded to the oxalate. Now, if we go back to what's in the blood, if everybody in the textbooks are right, and ionized calcium is the only calcium that matters, and we all know, because we've all been told at school even, that the homeostasis control system for calcium is incredibly good, and some of us can even name the hormones involved, you would think 4.5 billion years of evolution would control ionized calcium in the blood pretty well. Well, it controls total blood calcium exceedingly well. That is the top end of the normal range. That's the bottom end of the normal range. And this is total calcium before we put a horse on our supplement. This is same horses after our supplement. Nothing's really changed. We've dealt with some horses with horrible calcium problems with big head in Queensland and New South Wales, and we haven't had a horse probably any lower than that. They are all in the normal range. It does beg the question how the laboratories come up with normal ranges, but so total blood calcium very closely regulated. Ionized blood calcium, those of you that saw the magnesium thing yesterday, you'll see, that looks pretty similar in a way, in that we've got a whole range. So we've got horses with above the normal range, we've got horses below the normal range. When we put them on our supplement, some go up, some go down. In general, they go down. And you may say, oh wow, this is awful, isn't it? Look how many sick horses there are. Look at these. We've got 1, 2, 3, 4, 5. I've got 6 horses there that technically don't have enough ionized calcium in their systems. Is that important? This one here, which you can see, it started inside the normal range. This horse lives on the Shropshire-Wales border, which is about the lowest calcium area you'll find in Britain. So not surprisingly, its blood calcium, ionized calcium, is quite low. And we put it on our chelated calcium, and it crashed even lower. That one was not 55% ionized calcium, it was 46% ionized calcium. A couple of weeks after that blood test was taken, and with no change in its feed regime, it stayed on our calcium supplement. That horse was the best British horse at the Endurance World Championship. So don't tell me that it's actually really important that a horse's ionized blood calcium level are in the normal range. Normal does not mean optimum. And again, those of you that saw the magnesium thing yesterday, I've got a narrow range that I think is optimum. What's more interesting, and I haven't got the gory detail, but we can change this quite easily by adding chelated calcium. So what we believe happens is that either total calcium, so from here to here is tightly regulated, or the ionised and the complexed may be totally regulated. But certainly, there are certainly some textbooks that suggest that. What we think happens is if you feed your horse a whole pile of limestone or DCP or something like that, or it grazes a high calcium soil, it's going to have a lot of ionised calcium in it. And imagine that 55% goes to 57, and imagine that 5%, that doesn't make any difference, right? The horse isn't going to notice a difference between 55% and 57. But if that all gets squeezed out of the organically complex, and that 5% goes to 2% or 3%, that's a dramatic change in the amount of chelated calcium in its system. And our belief is that that is quite critical and quite important. I can't substantiate that, except that that seems to be the case. Our biggest problem is that there is no blood test to measure the chelated calcium. I can't ask for that. I can calculate it, and I sometimes get a negative number, which clearly isn't right. And that's why blood tests for calcium are useless to you. I can measure the change by making a calculation and seeing how that calculation changes before and after, but it doesn't give me an absolute number. So, now we get to the really tricky bit. What does chelated calcium do? We talked about what calcium ions do, and that's incredibly well studied, and a guy won the Nobel Prize last year for working all that out, and that's fantastic. But what does chelated calcium do? One. Okay. That's the truth. We have no idea. I mean, no idea, zero idea. Eloise, one of our equine science graduates, and I went to see the calcium specialist at the physiology and pharmacology department at Bristol University last year. We're talking about a department with 200 researchers in it. So we're talking about a big science-based department, and we said, this is what our product does. We have a whole list of things that the product do. Tell us how it does it. And he said, no idea. And then he got really expansive, because he said, if Britain allocated all of its science research budget to trying to answer your question, in 10 years' time, we probably still won't know the answer. So I am happy that I will go to my grave not being able to answer this question. But I'm a persistent sod, and I will keep trying to find out. What we have discovered is this. There are things called Vnecks. I put that pretty coloured chart up because that's how biochemists show this is a big protein molecule. Vnecks stands for voltage-dependent anion channels. Doesn't matter what that means. It's a diabolically bad name, even if you do know what it means, because they're not really anion channels at all. What's interesting about Vnecks is that they are capable of transporting large, uncharged molecules, and chelated calcium is a large, uncharged molecule. In other words, they're not actually transporting cations and anions separately. The most common one that they transport is ATP, which those of you with a biochemistry background will know is the primary energy source that your cells and your horse's cells are. This is the interesting bit. Even though these molecules are entire, Vnecks are capable of rejecting magnesium-containing chelated molecules and selecting for calcium-chelated molecules. They can actually select for some other things like strontium and a few other really, really mystical minerals. Getting a bit more biological now. Within all of your cells and your horse's cells are things called mitochondria. Billions of years ago we did a deal with some bacteria and said, you live in our cells and provide us with energy and we'll protect you from the world. And that's what mitochondria are. So they're basically bacteria that have been in our bodies for literally billions of years. And one of the things they do is store calcium ions. So I said that the inside of the cell was calcium-free, but there are storage organs in there, and mitochondria are one of those. They're located on the wall of the mitochondria, very close to an organelle called the endoplasmic reticulum. And the endoplasmic reticulum is another storage source of calcium ions. So it's interesting that they're located close to something which is dealing with calcium ions. But they're not moving calcium ions around, they're moving chelated calcium around. And finally, and this is to me a great surprise, Vnecks are the most common proteins in your cells. And when I say eukaryotic, that includes plant cells. When we talk about calcium signaling, we're talking about a biological technology that is almost ubiquitous. It's incredibly common. That is all we know about how chelated calcium works. All we know is that there are things in the cells that can manage the molecules that we're talking about, and that when we feed horses on chelated calcium, all sorts of wonderful things seem to happen. And we're learning more and more as we do more and more studies on horses with all sorts of problems. So, I'd love you to come and talk to us on our stand, which is down there in the far corner. I'm not sure whether no, I didn't. Look at our YouTube channel. We've got a magnesium video on our YouTube channel, and by about Monday or Tuesday we'll have a calcium video on there as well. It's not there quite yet. Anybody got any questions? I've finished in 40 minutes, so...
Chelated.
Okay. Okay, so the question was, if chelation is important, is chelated selenium, why is it important in selenium?
Is it important?
I think it is, actually. I think for most minerals, the chelation improves the absorption from the gut. And that is probably true of the calcium as well. For most minerals, though, and certainly magnesium, which is the only one I've studied, when the chelated mineral gets in there, it does separate out and release the ion. Now, chelated selenium is definitely safer than the sodium selenite that we were in Europe forced to use. We weren't allowed to use chelated. Now, Alltech have a selenium yeast product, don't they, which we can now use, but we weren't allowed to use that until 3 or 4 years ago, perhaps 5 or 6 years ago. So chelated minerals appear to be safer because not only can they be moved in easily, they can probably be excreted easier as well. But I think we're in the area of maybes and hopes and whatever. But I generally prefer using a chelate because I think they're safer.
Now, have you had any, from your results, any indications of excess calcium and any effect of the chelated calcium?
Okay, that's a very, very good question. And the answer is no. I think since 2009, when we started using chelated calcium, we would have had not a single excess. We've been selling this technology in our cage bird business all around the world, and I do mean there would be millions and millions of cage birds now consuming our chelated calcium, and no, we don't see any excesses. And I think the sorts of things where excesses appear, we're talking about gallstones, kidney stones, deformed bones. We're doing a trial on kissing the spine, for example. A lot of the vertebrae are misgrown and whatever. All the evidence is that chelated calcium, because it's enabling the osteoblasts and the osteoclasts to work properly, actually reduces the risk of all those things and even treats those problems. So no, we don't overdose. Having even a growing horse, more importantly in a growing horse. In fact, there are sort of three stages of a horse's life that are important. The lactating mare, the fast-growing horse, and with big head, they're the ones that show the bone deformations because their bones are very plastic and mobile. And the third group seems to be the horses that have reached mature size and are pumping calcium into their blood, into their bones rather, as part of the process of hardening them up. So they're going from soft bones to hard bones. And all of those have a huge calcium demand. And although we're not providing the mineralized calcium, we are enabling those processes to function better.
Setting the calcium phosphorus ratio.
Frankly, we probably don't give enough calcium necessarily to do that. So, for example, on our big head trials here, we pulled all of the limestone out, and our loading dose of calcium was half the amount of calcium that had been supplied traditionally, right? And then our maintenance dose is another third of that. So we're actually providing a lot less calcium. And now I know that having low ionised calcium doesn't seem important. I'm not convinced that we need to pump our horses full of limestone and whatever. The jury's still out on that. I think, you know, we need to study that better.
Where do you get your source of chelated calcium? Where does it come from?
Where does it come from? In Australia, I'm not 100% sure, but I'd be amazed if it's not China. Certain. Yeah, okay. Chelated calcium, there are a variety of chelated calciums. I'm not going to tell you which ones we use, but calcium gluconate, calcium citrate, calcium lactate. You can buy amino acid calcium. Our trials tell us that they all work. So there are actually quite a few things to choose. But we do have a patent, so if you wanted to do it yourself, you might get a letter from my lawyer. The question, did everybody hear the question?
No, not really.
Okay. The basic question is, did we control things like vitamin D levels and whatever? And the answer is no, and this is exactly why good science is very difficult, because there are so many variables, and a horse is an incredibly expensive, huge animal. You know, it's much cheaper to do it on 10,000 rats than a horse or mice or whatever. So no, we don't. But on our big head trials here, actually we did a lot more than normal. We did urine samples on them. We did a full panel of blood, different calciums that we could, the magnesium that we could. We had to use a lab at a hospital in Sydney, because we did PTH, we did calcitonium, we did vitamin D. And if you want to come to the stand, I can show you the results, because Big Head's upper name is hypoparathyroidism, and actually loads of the horses were not using parathyroid hormone to control their regulation. Their vitamin D levels were all over the place. So I'm not sure that it told us anything, but yes, we did do that in that case, but it was expensive. In Britain, I can get an ionised blood calcium test easily. I can get a parathyroid hormone test easily. Here, the parathyroid and the calcitonium, one went to a Melbourne hospital and one went to a Sydney hospital. And I think
they want you to stop because the next person's ready.
So, thank you very much indeed for your attention.